and welcome to the Arkansas Embray YouTube channel. I'm Stephanie, and I'm here with Axel from BigOmics Analytics, and I'm really excited to share with you about this software. I was first introduced to the BigOmics Analytics software while I was at the Frontline Genomics Festival of Genomics and Biodata Conference, and I really think that this is going to be a software where you can take your omics data and fast track your way to getting biological insights. So, welcome, Axel, and going to turn it over to you to give an introduction and a little demonstration of what this platform can do for us.
Yes, thank you very much, Stephanie. So I'm Axel Martinelli, and I'm the head of biology at BigOmics Analytics. And as Stephanie already mentioned, we are a Swiss-based company, and we develop a bioinformatics platform for the analysis of omics data. Today, in particular, I'm going to talk about the Omics Playground platform, which is dedicated to the analysis of transcriptomics and proteomics data. So without further ado, I'm going to start sharing my screen and showing what our platform is capable of. So here you have the main page of our platform. As you can see, it's a cloud-based solution which runs over a web browser, so you don't need to have any expensive hardware to run it. And the idea behind this platform is to make the visualization and interpretation of transcriptomics, including single-cell RNA-seq data and proteomics data, accessible and interactive to most users. So here in the main page, I have now several data sets that I already uploaded to the platform. I selected one just to showcase the main feature of the platform. As you can see, we have several topic areas. So I'm going to start quickly by going through the clustering analysis. So the platform covers some of the basic aspects of omics data analysis. So for example, we have dimensionality reduction in the shape of PCA, TSNE, or UMAP plots. You can see an example of the PCA plot. And most of these plots can be interacted with via setting menus, or there is a panel here with other options where you can, for example, select the visualization type of these dimensionality reduction plot. You can even turn them into 3D plots if you want, which are interactive. The platform also produces heat maps, of course, it's bread and butter. And here you can see an example of both a dynamic and a static heat map. Our platform will also produce different gene blocks that you can then annotate via a large collection of databases. We have, of course, your bread and butter differential gene expression analysis. So we produce volcano and MA plots, as well as more specific views about pairwise comparison that you can select here, and an overview across all the pairwise comparisons you have in the data set. We have, of course, tables which are interactive, and all the tables and figures can be downloaded. So figures can be downloaded as PNG, PDF, or you can download raw data as well as CSV files, whereas you can get the table data in a CSV format. We also have some more advanced features that you probably haven't considered before when doing omics data analysis. For example, we have a biomarker selection dedicated module, which you can see here. A simple example distinguishing glioblastoma versus healthy samples. And we run this based on different machine learning algorithms, which we combine. We have a gene set enrichment analysis tab, and here you can see an example of how we represent data. It's this typical GSEA plot. And for this kind of analysis, we use, as I mentioned, large collections, more than 30 different databases. You might recognize some of the popular ones, such as raw terms or hallmark collection, but we have also a lot more, for a total of more than 50,000 gene sets or pathways that you can use in your analysis. And still on the theme of gene set enrichment analysis, we also perform pathway analysis based on the wiki pathways, and Reactome databases, as well as producing some Go graphs. So here you can see, for example, an example of a Dickey pathway plot. Is an interactive plot, so you can see more in detail which genes are down regulated and in which component of the pathway, they affect. As part of the more advanced analysis, we can also do comparative analysis. So for example, we can query either all your uploaded pairwise comparisons, or alternatively, if you don't have many data sets, we also have a pre-uploaded collection of more than 6,000 geo dataset experiments, so more than 10,000 different pairwise comparisons that you can query to compare your selected pairwise comparisons with those and see which ones have the highest level of similarity. And then you can actually go into a more one-on-one type of analysis through the compare dataset platforms. Like here, you can select one of your uploaded datasets, and based on that, select a pairwise comparisons. And in this case, I'm showing you how we can actually use our platform to compare proteomics and transcriptomics dataset. So this is a transcriptomics dataset. I uploaded the corresponding proteomics dataset. You can see that the two plots here are pretty similar, showing that there is a high correlation between the two data types, which we can see more in detail when we look at the whole correlation plot. And then we can also go more in details by looking at individual genes or proteins, see the level of correlation or lack of it, if this is something that might be relevant from a biological point of view. And finally, when it comes to advanced feature, we also included the drug connectivity map, which is really useful if you are either trying to identify drugs that could be repurposed for a particular disease, or you're trying to understand the mode of action of a drug, or trying to identify a good partner drug for a novel compound that you might have developed. We have a self-profiling dedicated tab where you can consult more than 5 different databases, the extent of our databases to identify the nature of data set. It is more useful for single-cell RNA-seq data, but I sometimes use it also for bulk RNA-seq data. In this case, it correctly identifies the nature of my samples as brain samples. And finally, we also included double GCNA analysis, which is something that some biologists really find that it's supposed to be a lot of plots for this as well.
Yeah, there's really a lot of features in the software, like I said, so very excited to utilize it. One question that I know a lot of people have asked is what all organisms are supported in this software?
So that's a very good question. Right now, we have support for mouse, human, and rat data. However, as you can see here, we have actually working on the latest release of the platform that will include support for more than 120 species. Here you can see a quick list of all the species that we have. This includes some popular model organisms such as C. elegans,
Yeah,
Drosophila,
right.
and Saccharomyces. And in the coming future, we plan to release a version where you can basically upload any organisms, as long as you have a reference genome, of course.
Yeah, that's really exciting. I'm really looking forward to this. And so so if anybody has more questions about BigOmics, where can they find out more information and contact you?
Yeah.
Yeah.
So here we have some information on where you can contact us. We have our website, www.bigomics.ch. Otherwise, we also have a email address for general contact purposes. So it's hello@bigomics.ch. And we are definitely happy to answer any questions or requests you might have.
Ah, perfect. Yes, and if you're part of the Arkansas Embray, I would be your source of contact. You can email me at sbyrum, byrum, at ums.edu. And so this is going to be supported through the data science core. And I do have an example data set loaded into the software. So if you have more questions, I'll be happy to share with you how you can access the software. So please contact me with any other questions as well. So thank you, Axel, for meeting and going over this with everybody. And I'm looking forward to working with you further. And also, Axel does have a lot of videos already on more details about the different modules. So there is a lot more information on how to access and utilize this platform. This won't be just the only overview. So,
Absolutely.
yeah.
Absolutely. Visit our website and you can find most of the information there.
Yes, perfect. Well, thank you very much. I appreciate your time. So,
Thank you very much, Stephanie.
bye.